In this video, I'm going to review meiosis, Mendelian genetics, and non-Mendelian genetics. So first up is meiosis. I have here drawn a very oversimplified version of what happens in meiosis. You'll notice we start with a diploid cell. During interphase, the DNA replicates. Then, during meiosis 1, crossing over occurs where the genetic material from the paternal side and the maternal side mix together, and homologous chromosomes separate. Then in meiosis 2, sister chromatids separate. So we end up with haploid cells that have half of the starting number of chromosomes, and they are unique from each other. So as a reminder, in mitosis, we end up with genetically identical daughter cells. We end up with 2 of them, but in meiosis, in this, we end up with the genetically unique daughter cells, and we end up with 4 of them. So when the goal of meiosis is to produce gametes, so either egg cells or sperm cells, and then we learned in class that those egg cells and sperm cells come together to form a zygote, and that's what fertilization is. So I want to remind you and kind of give you an example. This isn't what happens after a mom and dad's genetic material comes together. This is like, for example, what happened in my body to produce an egg cell. And the red would represent my mom and the blue represents my dad, but this is not like right when I was becoming a human. This is like in my body to produce the egg cell that made the baby that I'm now carrying. And in my husband's body, he had a sperm cell that was half his mom's genetic material, half his dad's genetic material. And then these 2 gametes came together to form the baby that I'm now carrying. So my baby has some genetic material from my mom, some from my dad, and some from my husband's mom, and some from my husband's dad. So that's part of what leads to genetic diversity in our population and within our families is the uniqueness of the DNA combinations produced by crossing over, the mixing of the genes that we have in our body. And also, independent assortment also helps lead to genetic variety. So independent assortment is this idea that the chromosomes in our cells can sort independently. So, for example, if I have two chromosomes in my cells, chromosome 1 and chromosome 2. So I represented chromosome 1 as green and blue, chromosome 2 is orange and red. Say that orange and green were from my dad and blue and red were from my mom. Green and orange don't necessarily have to stay together. It could be that orange ends up with blue. So it could be that the blue and the reds end up together and the green and the oranges end up together. Or it could be that blue and orange ends up together and green and red end up together. Or some combination of that. So this is the idea of independent assortment. So our chromosomes sort independently of each other. So in humans, we have 23 chromosomes. So what that means is there's 2 to the 23rd possibilities, or 8 million possibilities of combinations of our chromosomes. So again, that leads to genetic diversity. So on the topic of Mendel's laws, we also have the law of segregation. And this is telling us that so say I only had 2 chromosomes again, so chromosome 1, green and blue, chromosome 2, orange and red. The law of segregation tells me that my 2 chromosome 1s are going to end up in separate cells from each other, so that each resulting gamete has 1 copy of each chromosome. So it's not as if one of the gametes is going to end up with 2 copies of chromosome 2 and 2 copies of chromosome 1. These chromosomes are going to be separate from each other. They're going to segregate. So 1 of each chromosome ending up in each gamete means 1 of each gene ends up in each gamete. So say, for example, the gene for hemoglobin were on chromosome 2. The gametes that are made are not going to have 2 copies of chromosome 2 with 2 copies of that hemoglobin, because that would mean that some of the gametes that are made wouldn't have the hemoglobin gene. So each resulting gamete has one of each chromosomes in it. So that way, offspring have all 23 chromosomes from parent 1 and all 23 chromosomes from parent 2. And so it's not, the two genes and the traits from the parent are not sticking together. Okay, so in Mendelian genetics, that's when we practiced Punnett squares the most. So I'm just going to kind of do two quick examples. In the first example, I have a homozygous recessive individual crossed with a heterozygous individual. Remember, homozygous recessive would be two little letters. That's how we'd represent it. Heterozygous would be a big and a little. So I set up my Punnett square where I have my two gametes from this individual and my two gametes from this individual and completed it. And I ended up with 50% of the potential offspring having the dominant phenotype because they're big R, little r, and 50% of the offspring having the recessive phenotype, little r, little r. So this is a 1-to-1 ratio, right? Equal parts possible of dominant phenotype, equal parts possible of recessive phenotype. In this down here, what we call a dihybrid cross, where we're looking at more than one gene, I have a homozygous recessive individual crossed with a heterozygous individual for two traits. C is the type of hair, and D is the color of the hair. So big C's curly hair, little c straight hair, big D's dark hair, little d's light hair. And this is an oversimplification because our hair color and hair type don't come from just one gene, but this is kind of just for an example. So I set up my dihybrid cross here. First, I did the easy one that's all the same, little c, little c, little d, little d. The only possible gametes from there are just little c and little d. So here we have little c, little d. This one's a little bit trickier. I have to think about all the ways that these could be combined where there's one c and one d. So it could be big C, big D, big C, little d, little c, big D, or little c, little d. So then I filled out my dihybrid cross, my Punnett square, and I used different colors to see what the different genotypes that I ended up with, and I realized that my phenotypes are 25% curly and dark, that's dominant, 25% curly and light, dominant for C, recessive for D, 25% straight and dark, it's recessive for C, dominant for D, 25% straight and light, which is recessive for both. So you can see that 1/4, 1/4, 1/4, 1/4 is my breakdown. So this would mean I have a ratio of 1-to-1 to 1-to-1 because they're all in equal parts. Okay, and now for some non-Mendelian genetics reminders. There's codominance, where if there's a light and dark parent, we end up with spots. Codominance, like coworkers, they both show up. Incomplete dominance, light and dark makes kind of like a gray or a blending in of the color. Or the trait multiple alleles is like, for example, with blood type, there's more than just dominant and recessive. There's A, B, and O. And if there's a blood type individual crossed with an AB blood type individual, that could mean that A blood type could be AO or it could be AA. So I set up both just to show you. AO crossed with AB versus AA crossed with AB. O is recessive, so we don't say it if A is present. That's why it could be AO. And the resulting distribution of phenotypes for this one is 1-to-2-to-1. A blood type 1, A blood type is the 2. There's 2 possible offspring with A blood type. AB is just 1, BO is just 1, so 1-to-2-to-1. In this one, it's A or AB, 1-to-1 equal parts. X-linked is when the chromosome is on the X, when the gene is on the X chromosome only. For example, if a dad is colorblind and a mom is a carrier, so I have to write X's and Y's, and I put the gene as a little c here and fill this out. And the result here is a 1-to-1-to-1 ratio again. There's also polygenic, where multiple genes influence one trait, and pleiotropy, which is where one gene influences many traits. And as a reminder, environment can impact the expression of genes too. Like, for example, there's a flower that, depending on the acidity of the soil, will be different colors.